we have enough time to get through the program for today. So thank you, everyone. Good morning and welcome to another Tuesday morning cardiovascular grand rounds. Just a few housekeeping announcements before we get started. We're really now in the process of nominating speakers and trying to get the schedule set for the next academic year. So I do encourage you all, if you have, speakers that you would like to see on the grand round stage, and keeping in mind that we will be trying to transition back to mostly in-person meetings in the next academic year, please feel free to reach out to Liz Reitman, to Rohan Kira, or myself with your nominations. We do have a nominations form that we'll put in the chat that you can use as well. Just same housekeeping things that we've always known. Ask questions either in the chat or using the hand raise function. We're hoping that this session, which is a review of some studies from the recent ACC meeting, will be one that is quite interactive and one that we can ask quite a number of questions in. So with that said, and just sort of one more update, next week we have Karen Stout on May 17th and Marma Vasegi on May 24th. So those are some talks to look forward to. So with that, we'll get started. We have two talks today. We'll start off with Lama Ghazi, who's presenting really a homegrown study that we're all aware of and really very proud of to have seen on the big stage at the ACC. So she'll be presenting the PROMPT-HF cluster randomized trial, and then that will be followed by Tony Giaimo presenting the Horizon trial. So to get started with our first speaker is Lama Ghazi. She's an MD PhD, postdoctoral fellow at the Clinical and Translational Research Accelerator. She received her bachelor's degree in biology and medical degree from the American University of Beirut in Lebanon. She then completed a 2-year postdoctoral fellowship in vascular biology at the University of Alabama in Birmingham, and then received a PhD in epidemiology before joining Yale in 2020. Lama has been actually someone that I'm starting some collaborations with. She's an active member of the American Society of Nephrology and the AHA. Her interests really are in chronic disease prevention and management, specifically in hypertension, data science, cardiovascular diseases, and chronic kidney disease. She's been a collaborator with Tariq, Nihar, Perry on the PROMPT-HF study. So with that, I will turn this over to Lama. Thank you.
Thanks. Just making sure everyone can hear me, and I'll start. So I'll go over our study here on pragmatic trial of messaging to providers about treatment of heart failure. It's a cluster randomized trial, and happy to have been part of it over the past 2 years. A lot of authors are here, and I'll skip to the funding information and disclosures. Basically, it was funded by AstraZeneca, but they had nothing to do with the analyses or the results. And I'll go over the outline. I'll first start with some background, objective methods, and results. So I know most people here are experts in the field. You all know that HFrEF is a complicated disease with worse morbidity and mortality than most cancers. And there have been many advances over the past 35 years in drugs, as well as device-based therapies for management of heart failure with reduced ejection fraction. And I'd just like to point out, you have ACE, ARB, ARNIs, beta blockers, MRAs, and most recently SGLT2 inhibitors, which are now considered the foundational quadruple therapy for HFrEF management, as by the most recent also 2022 AHA guidelines. Those came out after the study completion. I'd just like to note that it is recommended to start all 4 classes of GDMT, and medications may be simultaneously started at the initial dose recommended for HFrEF, or they can be started sequentially without the need to achieve target dose before initiating the next medication. And in fact, use of all 4 drug classes has been estimated to reduce all-cause mortality by around 73% compared to no treatment. Now, despite all of that, and even the fact that a lot of these medications have shown more than 2 decades of benefits, the uptake and use of these therapies have been subpar. So the CHAMP Heart Failure Registry, which consists of 150 primary care and cardiology practices across the US, between 2015 and 2017, they looked at baseline uptake basically of GDMT, but in this case, only the 3 medications were recommended before SGLT2 inhibitors were on the market, and then afterwards, 12 months after, and that's the orange bar. So for beta blockers, as you can see, around 80 to 82% prevalence of use of beta blockers, ACE and ARBs around 66 to 62%, MRAs around the 34% range, and ARNIs around the 14, and it increased to 20%. Now, there are a lot of reasons for lack of improvement in GDMT uptake. I'll be focusing more on the right side here. So there are patient reasons, provider reasons, as well as system and payer causes. So on the patient side, some contraindications and side effects. System side, as well as payer, it includes the high cost, inadequate access, and whatnot. And in our case, we specifically focused on the provider side, which is to look at ways to overcome inertia, knowledge gap, as well as maybe inadequate follow-up of care. Now, there have been previous efforts to improve heart failure with reduced HFrEF outcomes in the past few years, using or leveraging as much as we can of technology. So the CONNECT heart failure study was done at Duke. It was a cluster randomized clinical trial, and they evaluated the effect of hospital and post-discharge quality improvement intervention on HFrEF outcomes and care. They showed there was no significant difference in HFrEF rehospitalization or quality of care score. The PACT-HF study was done at was done at Ontario in Canada, and here they evaluated the effectiveness of implementing group of evidence-informed transitional care services. These include education, referral systems, quality of life assessments among patients discharged after hospitalization for HFrEF. And they also showed no improvement in readmission, ED, or death at 3 months. I'd like to note these are very resource-intensive efforts that did not yield positive results. And there has been also the EPIC-HF randomized clinical trial in which they checked or investigated the effect of a patient activation tool, which consisted of a 3-minute video and 1 patient medication checklist that was delivered electronically to the patients before their cardiology visit, 1 week prior, 3 days prior, and 24 hours prior their cardiology visit. And in this case, there was an increase in GDMT uptake. Here it consisted of initiation or intensification of ACE-ARB ARNIs, beta blockers, and MRA. However, in this case, not only did they not look at SGLT2 inhibitors, it was patient-facing, as well as this is difficult to implement on the large scale. Suffice to say, this made us think about how we could leverage the EHR to overcome the barriers to increase initiation of GDMTs, especially when it comes to clinical inertia, lack of knowledge, and understanding what medications are in the market. So electronic health records, embedded clinical decision support via best practice alerts can be used to target and individualize GDMT recommendation. This approach is easily scalable, and it is a low-cost way to accelerate high-value care. And this would be a good segue to start with the objective of the PROMPT-HF trial. So again, this is a pragmatic trial of messaging to providers about outpatient treatment of HFrEF. It was designed to test the hypothesis that timely and targeted alerting of recommendations about medical treatment of HFrEF tailored to the individual patient would lead to higher rates of GDMT prescription when compared to usual care. So this study was a clustered randomized study. We randomized at the level of the provider. We recruited 100 providers that are high-yield providers at Yale that saw HFrEF heart failure patients. Those providers were either randomized to receive an alert or no alert. Now, the alert popped up to these providers if the patient was eligible to receive an additional GDMT. So eligibility was based on the fact that the patient was older than 18, their left ventricular ejection fraction was less than 40%, and they were not on all 4 GDMT therapies. Now, we collected data via EHR, and our primary outcome is an increase in number of prescribed GDMT at 30 days. I'll show the details of the alert. So in order to test the alert, we actually had multiple focus groups with a lot of cardiologists here at Yale in order to make sure that this was fulfilled basically the 5 pillars of CDS, or clinical decision support, which is making sure that the information, the recipient, the intervention, the format, as well as the workflow are ideal in this case. So if the patient is eligible and they see a provider that was randomized to the alert, what they see is basically the first statement states that the patient meets the criteria for HFrEF, and these values are presented. Their most recent ejection fraction, their blood pressure, potassium, heart rate, and eGFR. Now, as I mentioned, there are 4 different classes. For each of these classes, if the patient was on them, it would be grayed out. So for example, for this patient, that patient was on Entresto. So this is why you see the class listed above, and it will be grayed out in which medication along with the dose. For the remaining medications, they were not on them. So this is why they appear in red. So they're not on beta blocker. However, there is a contraindication. In this case, their heart rate is lower than 60. They're not on MRA, and it will list the contraindications if there are any, including allergies as well. So in this case, MRAs are contraindicated because the potassium is 6, so greater than 5. It fulfills that first exclusion criteria here. However, they're not on SGLT2 inhibitors, and there are no contraindications or allergies reported. Following that, there is a link here that would take all the provider. It'll take them actually to our website, the PromptHF website. There is a summary of the protocol, the guidelines, and all the evidence in there. Here, this is a modifiable open order set. I'll get into that in a bit, in which the medications that are recommended for the patient would appear. And there is an acknowledge reason on the bottom in which the provider can click either I'll adjust medications, med changes not clinically indicated, or remind me in 2 days. This is the complete order set. So this is not the way it appears for the patient I just presented. For that patient, the only part that will appear is the bottom part, the SGLT2 inhibitors. I'd just like to note that they do appear in alphabetical order, each class and the medications available. Again, our primary outcome is the addition of GDMT class. So if a patient started on 0 medications, 0 classes, but went to having 1, 2, 3, or 4, that would be considered a win. So they actually experienced an outcome. If they went from 1 to 2 and 3 and 4, and so on, that's also considered a win. However, if they stayed on the same number of medications or there was a decrease, we just consider them not to have experienced the outcome. Just to note, for instance, if they were on ARB and MRA and they want to be on an ARB and SGLT2 inhibitor, as you can see, there is no increase in number of medications. So in this case, we do not consider them to have experienced the outcome. However, if they were on ACE inhibitors and then SGLT2 and beta blockers were added, they were considered to have experienced the outcome. So our sample size consisted of 1310 patients to achieve a 91% power to detect a 10% difference between study arms, so alert compared to no alert, at alpha 0.05. Our primary outcome examined the association between intervention and outcomes using generalized linear models adjusted for pre-specified baseline characteristics, and we accounted for clustering at the provider level. And statistical significance for the primary outcome was based on a p-value less than 0.4048, and this was after accounting for 2 interim analyses. We had 2 DSMB meetings at 25% and 50% recruitment. Just like to thank Tariq, I did use a couple of his slides that he used at ACC here. So this is an embedded EHR pragmatic trial, and here I just want to show the hospitals and the clinics that were included. I'd like to note that the study actually started in April 2021, and we completed recruitment in October 2021. So over approximately a 7-month period, we completed a study, which is quite surreal and very exciting to be able to conduct a study that fast. Now for the exciting part, I'll just go over some baseline patient characteristics in the alert and no alert group. So as you can see here, the median age of patients was 72, 30% of them were female, and that reflects the disease itself. 70 to 19% were Black, around 9% were Hispanic or Latino, 85 to 86% were on Medicaid or Medicare. The vitals at the time that the alert fired, you can see here, their blood pressure was well controlled, 120/70, their heart rate around 74, their kidney function was pretty good, their eGFR was 56.9, 58.4, that's the median, and ejection fraction was 32, the median was 32%. A few comorbidities listed here. As expected, most of these patients have hypertension, 85% of them, obesity in 21%, diabetes in 38%. Now, when we look at the baseline GDMT uptake or that patients were on, 85 to 83% were on beta blockers, around 70% were on ACE, ARB, and ARNIs, around 28 to 30% on MRAs, and around 10 to 12% on SGLT2 inhibitors. I'd like to note that this study took place while already at Yale there was increased effort to improve SGLT2 inhibitors sort of prescription and uptake. Now, I'd just like to compare our baseline characteristics to the CHAMP-HF study. Again, this is a registry of heart failure patients. And as you can see, our population is pretty representative of the general population in CHAMP-HF. So we have a pretty good presentation, both of age, the demographics, as well as the medication use. Now, for the main results. So I'd just like to orient everyone. Blue is patients no alert, and purple is the alert, and these represent the % increase from baseline. So after 30 days from the alert itself, what we see here is that patients who were randomized to the no alert group, 18.7% had an increase in GDMT, so an additional class of GDMT, compared to 26% approximately in those who were randomized to receive the alert. This was statistically significant with a p-value of 0.03. Our risk ratio was 1.41. And I'd like to point out that the number of patients for which an alert was needed to result in an increase in an addition of GDMT class was 14 in this case. We were not powered to look at individual level medication classes. However, you can see here the trend is pretty much similar, with those being in the alert group having an increase in each of these classes more than the patients that their provider was randomized basically to the no alert group for all of them. Now, these were our pre-specified subgroup analyses, which looked at basically age cutoffs, sex, race, their ejection fraction, whether it was less or more than 20%, whether they were seen by a cardiology or a non-cardiology specialist, whether they were on Medicare or Medicaid or other types of insurance, and the number of GDMT at baseline. Overall, you can see here the interaction p-value was not significant for any of them. And again, our overall risk ratio, our effect was showed that there is a 40% increase in ordering an additional GDMT class with the alert compared to the no alert group. And here our secondary endpoint, and again, not our primary endpoint, we look not only at an increase in GDMT class, but increase in dose of each of these classes. And in this case also, if we look at all GDMT, 36.2% of patients randomized or in the alert group had an increase in GDMT class or an increase in dose compared to 26.2% of those in the no alert group. And this was also statistically significant. And in this case, the number of patients needed to alert to observe an increase in one GDMT class, was 10 in this case. Similar to when we looked at only increase in GDMT class, when we looked at each of these individuals' classes, you can see those again randomized to the alert or those in the alert group in this case had a greater increase in each of these classes over 30 days compared to those in the no alert group. However, when we looked at secondary outcomes, and here ED visits not resulting in hospitalizations, ED visits resulting in hospitalization, direct hospitalization or death, there was no significant difference between the alert and no alert group, and the rates were not that high. And this is expected because the follow-up here is also only for 30 days. We did assess provider feedback. So we did send a REDCap survey for all providers who were randomized to receive an alert, and 79% of them agreed or strongly agreed that it was effective at enabling improved prescription of GDMT for patients with HFrEF. Now, I don't know if you remember, but on the bottom of the alert, there is the acknowledge section. So based on our findings, 25% of the providers accepted the recommendation. 48% indicated they will address the suggested intervention in the future, and 14% indicated that patients do not meet the criteria. If providers either click that they will address either the patient does not meet the criteria, what we had was a free text option in there, and most of the reasons for not prescribing the GDMT was either hypotension, renal failure, decompensation, or medication not tolerated, or patient refused. So the strength of this study, it is entirely embedded, pragmatic clinical trial. The characteristics of the patients, as I showed, reflect national cohorts. We enrolled and finished the study in less than 1 year, and the alert provided specific guidance-based information and not just information alone. So it was an actionable alert. There are several limitations, and this includes the fact that this is a single academic health system in which the study took place. We only included high volume providers, those 100 providers that I mentioned in the beginning that saw HFrEF patients within the Yale healthcare system. We tested in the outpatient setting. Just a caveat here, we are running a similar trial in the inpatient setting, and we're excited to share the results when the trial finishes. We also, the alert was tested in EPIC EHR ecosphere, so it would need some adjustment to transfer to other sort of health systems such as Cerner. There is an increase in dose was a secondary outcome, and so far we only have, we looked at the impact at 30 days, however, not beyond that, but these analyses are upcoming. Finally, to sum up, this is a personalized best practice alert that triggered via the EHR during office visits, led to significantly higher number of patients on appropriate GDMT. It is a relatively low cost and widely scalable intervention that can be part of a multifaceted program that improves the quality of care in patients with HFrEF. There is a first avenue to start increasing GDMT uptake, and we are excited to see how this can improve and be scalable to other healthcare systems as well. Finally, I'd like to thank the participants of Prompt, as well as Perry, Tarek, and Nihar for allowing me to be part of this when I started my postdoc. And if anyone has any questions or comments, more than happy to answer. Thank you.
Lama, thank you so much. That was excellent. And I think it's just, really amazing to see, all the work that, you guys have done, and really to, know that this is a, homegrown program, and really excited to see the different iterations, of the prompt enterprise that arise from this. I think this is a good point to, stop and take questions and discuss the study before we move on, to the next one. So if anyone has, questions, I think that this is a, good time to ask those. Maybe I'll get started. So I think this is excellent. One thing that you did show, when you showed your table one, is that a number of these patients have significant comorbidities, right? And, given the way things are going, or what is your thought or the planning around how to address this, kind of in a more comprehensive and, holistic way? So do we embed multiple, recommendations in the same prompt? Do we send different, all right, here's your heart failure one, here's your blood pressure one, here's your diabetes one. But, how do you balance kind of the comprehensiveness of the intervention versus, diluting the effect of the intervention, given how, targeted and precise your recommendations are. So it's kind of that tension between being more comprehensive, but, if you send individual, alerts, we're just going to end up in, alert fatigue.
Yeah, absolutely. Great question, especially that a lot of patients have hypertension and diabetes along with HFrEF. The problem is a lot of pushback that we've seen is a lot of providers, if they're not their primary care physicians, they push sort of the recommendation or the change in medications to a specialist, and the specialist sometimes push it on the primary care provider. So honestly, this requires another study just to see, or maybe some focus groups just to see what is most acceptable by clinicians and would not lead to alert fatigue. I won't be able to give an answer right now, but I don't know how I think one of the reasons we saw success with this study was that it was targeted and there was an actionable item or actionable sort of item to change. So changing medications, maybe joining diabetes and HFrEF would be ideal right now with SGLT2 inhibitors. But yeah, I think that would take another study to be able to answer that. But thank you.
Other questions? There are none. I'll, keep sort of taking the liberty to ask more. Tarek, I see you're on. I might ask you to comment. So should we all be expecting to see some, prompt alerts in our inbox soon? What are the next steps in terms of being able to, roll this out more widely? Are you still in study mode? Or are you ready for more widespread implementation?
So, Yeri, that's a really good question. So we're studying this in the inpatient setting as well, where, as you know, most clinicians are bombarded with best practice alerts. It may not work in the inpatient setting, so that would be important to know, because, what Terry and Nihar and I have really pushed for is that we need to study all these interventions before we launch them. Unfortunately, they're launched across the EHR without any study, and then they just kind of stay there. So we're going to do this in the inpatient setting. Nihar is doing a study on lipids across the healthcare system. But the next step, as you alluded to, would be to make the alert much more intelligent. So it has to be, even more kind of specific to each individual patient, taking into account their renal function, their other comorbid conditions, and maybe more even comprehensive. Like if they have AFib, there's a recommendation for involvement of the EP team, for example. So this is just the first step. I think we need to kind of move towards making these a lot more intelligent.
Thanks, Tarek. And again, just really excellent work. I think we're all, very proud of the work you guys have done, and, looking forward to see what comes up next. Going to switch tack here onto the next study that we're discussing, but just to say that the floor is still open, Lama's still here. So if there are any questions, please feel free to put them in the chat, and we can circle back at the end of the next talk to address those. So with that, we'll just go ahead and move on to the next study. The next study is going to be presented by Tony Giaimo, who we all know well, but he doesn't need a lot of introduction to us. But Tony is one of our fellows, soon to be faculty in a couple of months. So congratulations, Tony. Tony did his undergraduate work at Fordham University and then spent the next 2 years teaching middle school science, which is probably the most difficult job he's ever had and will ever have, with Teach for America, before starting medical school at the New York Medical College. He joined Yale for residency in the primary care program, went on to become chief resident, and then started cardiology fellowship after that, after spending a year as a hypertension fellow. Tony is in his final few months of cardiology fellowship. His interests are in hypertension and prevention, and we're particularly excited, especially also in the prevention group, that Tony will be joining us as faculty starting in July. Today, Tony will be presenting the Apollo study, I think sort of really highlighting something that I think is coming down the pike and is on the horizon for most of us. And we thought it would be an important time really to start this conversation. So with that, Tony, I'd let you take the floor. Thanks.
Thank you for the introduction, Yuri, and thanks for the invitation to talk about the Apollo trial, which was presented as a late-breaking trial at ACC. Please let me know if at any point you can't hear me well. So the Apollo trial was looking at a small interfering RNA, SLN360, targeting Lp(a). And so I have no disclosures to report, and I did not participate in the trial. So Apollo was a phase 1 trial, and as you know, phase 1 trials are done to evaluate for safety and efficacy of the drug. And they asked the question, is a single dose of a subcutaneous injection of a short interfering RNA targeting hepatic production of apolipoprotein A safe and effective for lowering Lp(a) plasma concentrations? And so it's a little bit unusual to hear about a phase 1 trial in a late-breaking presentation, and then for us also to be talking about it here at Grand Rounds. But there's good reason for that. So let's just discuss the background of Lp(a) a bit, and why some people call treatment of Lp(a) sort of the holy grail of lipid therapies. So lipoprotein A is a cholesterol-carrying lipoprotein, similar to LDL, except that it has an apolipoprotein A covalently attached to the apolipoprotein B100, and I'll show you an image to clarify that. It's been known for some time that it has a very long circulating half-life, and you can imagine that that would contribute to its atherogenicity. It's thought to have such a long half-life because the apo-A interferes with the Lp(a)'s interaction with the LDL receptor, preventing, efficient clearance. And, quite interestingly, Lp(a) levels are genetically determined and not affected by diet or lifestyle. And so for a long time, we've known that when patients have elevated levels, there's not much that brings them down. But Lp(a), because it's an ApoB containing lipoprotein, can accumulate oxidized phospholipids and cause atherosclerosis. And there are different isoforms of the apolipoprotein A, and some have more or less atherogenic potential, and we won't get into that today. But importantly, transcription of the LPA gene to eventually lead to the production of apolipoprotein A is the rate-limiting step in the synthesis of Lp(a), and so that's what these drug developer was targeting, this rate-limiting step. And so I like this image because it shows the difference between an LDL particle, something that we think about and treat every day, and the Lp(a) particle. And you can see again that they both have an ApoB core, and the Lp(a) has this apolipoprotein A moiety attached to it, which causes all kinds of problems. But you also see that Lp(a) contains some low-density lipoprotein. And so, for some time now, we've known that Lp(a) is an independent risk factor for atherosclerotic cardiovascular disease, and is also known to be associated with the development of aortic stenosis, the progression of aortic stenosis, and the need for valve replacement. Sort of Mendelian risk studies have shown that the risk for ASCVD increases in a log linear fashion for levels that are greater than 20 nanomoles per liter. And we'll touch a bit on what is very high and what is not. And so this translates into an 11% increase in the risk for ASCVD for every 50 nanomolar per liter increase in Lp(a). And you can imagine, as is shown in this graphic here, from a study looking at the potential for treating patients as secondary prevention when they have high lipoprotein(a). There's patients with levels as high as 400 nanomoles per liter. And so you can imagine that their risk is extremely high. And there are some estimates that, there's over 1 billion people with elevated levels of Lp(a). But that's, again, considering elevation as starting at 20. And so what is the clinical significance of Lp(a) and why is it something that's received so much attention? And recently the European Society of Cardiology recommended that we should check Lp(a) at least once in any patient at cardiovascular risk. The AHA ACC does not go that far, but tells us that if we do check and the Lp(a) is greater than 125 nanomoles per liter, we should consider that a risk enhancing factor and more aggressively treat with statins or treating other, components of the patient's risk. The National Lipid Association, importantly recommends that we check Lp(a) in patients with a personal or family history of premature ASCVD or those with suspected familial hyperlipidemia because it may be the causal mechanism for their disease. And this comes from the fact that we now know that patients with extremely elevated Lp(a), we're talking about levels 300, 350 nanomoles per liter, have a cardiovascular lifetime risk similar to FH patients. So niacin and PCSK9 inhibitors are known to moderately lower Lp(a), but they've never been tested specifically for that purpose, nor shown to have a clinical benefit. And as I mentioned, there's an association with aortic stenosis. And so importantly, there is not a drug or a pharmacotherapy approved for lowering Lp(a), and hence, there's never been any proof that lowering Lp(a) lowers cardiovascular risk, but optimism is high, as you can imagine. And so, this drug is a small interfering RNA. I just wanted to touch quickly on the mechanism of how that works. We are somewhat familiar with this mechanism because we, recently had FDA approval of inclisiran, which is a small interfering RNA to prevent the translation of PCSK9. And so if you bring your attention down here where the drug is pictured on the bottom, you see that there are two strands as a component of this small interfering RNA, one a passenger strand and the other a guide strand. Once they enter the hepatocyte, they separate, and the guide strand then links itself to the mRNA that's been transcribed from the LPA gene. Once the guide strand binds that mRNA, the mRNA is then degraded, and the guide strand is left behind to then bind another mRNA. And it goes on to do that. We, in fact, don't know how long it continues to do that. This study went out to 150 days, and we know that it was still active at that time. And all of this is obviously to prevent the translation into ApoA, which is, as I mentioned, the rate-limiting step in the production of Lp(a). And so this study, again, a phase 1 trial, included patients 18 or older with no known atherosclerotic cardiovascular disease, but with an Lp(a) level greater than or equal to 150 nanomoles per liter, which is considered pretty high, and a BMI of 18 to 45. And so the exclusion criteria, I listed some of them here, but basically they excluded any patient with any significant illness. This was a phase 1 trial looking, primarily at safety and whether this drug can lower levels. So you can see the outcomes here on the bottom, looking for any safety signals, and then effect on concentration from baseline to 150 days. Secondarily, they looked at the effect on LDL and ApoB, and we can talk about that a bit. So this was a single ascending dose study. And so there were 8 participants per cohort, and there were 4 different doses of the drug provided, but to different cohorts of patients. And so the first cohort received 30 milligrams, and then they waited some time, they were observed, they were monitored, and then after a certain amount of time passed, they recruited a second cohort to whom they gave 100 milligram dose. And you can see then that they followed that with 300 milligrams and 600 milligrams. Each cohort was followed out for 150 days. And that's the data that was presented to us at ACC and in Giaimo. The 300 milligram and 600 milligram cohorts are still being followed out to 365 days to see durability and safety of the drug, but we don't have those data yet. And so here is the figure 1 for the trial. They screened 71 patients. As you can see, 38 were excluded, the majority because they had comorbidities or lipoprotein(a) levels that were less than 150. And the rest of the patients were randomized to one of the 4 treatment drug doses or placebo. And this was done at 6 sites around the world in the US, UK, Australia, and the Netherlands. And so this is table 1 showing the baseline characteristics of the patients that were enrolled. And in summary, these were young, patients that were 40 to 50 years old, otherwise healthy, with high Lp(a) levels. You can see here pretty even split between men and women. Almost no one had diabetes. A handful of patients had hypertension, and the lipoprotein(a) levels range from 142 to 338. And there were no Asian patients included, but there was a good mix of African American and white patients. And so in table 2, they report to us an assessment of adverse events. So importantly, they did not attribute any serious adverse events to the drug. You can see here in the 30 milligram dose group, there is 1 patient who's noted to have had a serious adverse event, which we're told was investigated and thought to be related to liver injury after receiving the COVID vaccine. That being said, 2 things I will point out: nearly all the patients had injection site reactions, especially in the 600 milligram group. 4 of those 6 patients had grade 2 reactions. And in that 600 milligram group and thought related to the injection site reactions was an increase in neutrophil count and CRP that the authors told us in their presentation was a transient increase. We're not told how transient. And they theorized again that it's related to the injection site reaction. And so here are the results. And so on the top, we have a graph that's showing us on the y-axis lipoprotein(a) concentration levels in nanomoles per liter. On the x-axis is visit. And here you can see that with each of the doses of the drug, there's really a robust lowering of Lp(a) plasma concentrations with, and I wrote it here, the 300 milligram dose led to a 96% reduction of Lp(a) levels at peak. And out to 150 days, you can see that the 300 milligram dose is still 70% lower plasma levels. The change in lipoprotein percentage is shown in the lower graph. And the 600 milligram, as the highest dose provided, led to a 98% reduction of Lp(a) at peak and 81% out to 150 days. And again, we will eventually have data out to 365 days to see how durable and safe this lowering was. And so the authors also looked at lowering of LDL and ApoB, and those are results shown in the graphs here. The top graph is LDL lowering percentage, and the bottom graph is ApoB lowering percentage. And you can see that in the 300 and 600 milligram doses, they received about a 21 to 26% reduction, the mechanism of which we can discuss. And so some limitations of this study: obviously, this is a phase 1 trial. Really, the authors described this as the first in human trial. which is sort of true. They were doing this at the same time as another drug company, and we'll talk about that. So this was a small cohort of very healthy patients without ASCVD. There was, for such a small cohort, we should look at, any safety signals. So there were elevations in CRP and neutrophil count with the highest dose. We're told those are transient. We don't know how transient, and they are of unclear clinical significance. As I mentioned, no hard outcomes as a phase 1 trial. And importantly, only a single dose was administered. And so this same group is doing a multi-dose trial as well. So we'll eventually have that data as they move into phase 2. And then the longevity of the effect is still to be determined. So this received a lot of attention, and for good reason. But I will point out that there is another drug with the same mechanism called olpasiran, which is a small interfering RNA that's targeting the same LPA gene transcription. In the Ocean A dose study, these phase 1 results were published in Nature Medicine just a few months ago. And the graphs here on the bottom are showing the results of their phase 1 data, in which on the y-axis we're seeing a mean % change from baseline in Lp(a) plasma concentrations. And you can see that this drug showed the same extremely robust lowering of Lp(a) nearly 100% at the highest doses, and it starts to gradually increase. They provided us data out to 225 days. So you can see here that the highest dose of 225 milligrams at an almost 100% reduction, and out to 225 days is still Lp(a) plasma concentrations are down by 80%. So olpasiran is in phase 2 trials already. They're enrolling, and we hope to have their phase 2 data in a year or two from now. And so that brings us to the drug that is much farther along, and that's pelacarsen, which some of you may have heard about. Pelacarsen is not a small interfering RNA, but an antisense oligonucleotide against the same target. At the highest dose of 20 milligrams sub-Q weekly, the phase 2 trial results, which are pictured here, showed an 80% reduction in plasma concentrations of Lp(a). And so with that robust lowering, they moved to phase 3, and the Lp(a) Horizon trial is currently underway, testing pelacarsen 80 milligrams sub-Q monthly versus placebo. It's a randomized double-blind placebo-controlled trial assessing the impact of Lp(a) lowering in patients who've already had atherosclerotic cardiovascular disease. And so they're looking at MACE in patients with an Lp(a) level greater than or equal to 175 nanomoles per liter. So a secondary prevention trial, exactly what we need. And they are targeting enrollment of 7,600 patients. And as far as I could find, they're already 50% enrolled. So we're expecting outcomes in 2025. So in summary, the Apollo trial, reinforced that Lp(a) is a clinically significant risk factor for atherosclerotic cardiovascular disease, and it's not currently modifiable, which is why they are in developing this drug. And so a single dose of siRNA SLN 360 that was administered subcutaneously is safe and effective at lowering Lp(a) in a small cohort of healthy patients with elevated Lp(a) plasma concentrations over 150. It is one of several RNA targeting therapies for lowering Lp(a), and as I mentioned, studies are ongoing to establish a reduction of Lp(a) as a beneficial target for the prevention of atherosclerotic cardiovascular disease. So I will pause there and see if anyone has any questions. Thank you very much again for the opportunity to talk to you about this study.
Tony, thank you so much. That was an excellent presentation, and I think one that we think is very relevant, obviously, in terms of being able to keep an eye on these important, developments kind of in our landscape, and, also a chance to start thinking about getting involved in some of these studies, if they're relevant, or in the clinical rollout when it comes to that. we'll take questions, but, maybe I'll start again, seeing as I'm asking lots of questions today. Tony, I think, one direct question for you. We know where we are in terms of the, landscape for LPA. What are kind of the direct clinical implications to us? So if someone's thinking, all right, this LPA story sounds, LP(a) story sounds interesting. What does that mean in our clinical practice now? What do you do, or what should we be doing now? And, I can later share kind of my experience, or, Erica, if you're on, you can jump in as well.
Yeah, thanks for the question. I did have a slide on that. I wasn't sure how much time I would have, but I'll show my clinical implications slide here. So, importantly, I'll start by saying that lipoprotein(a) contains LDL. And so when we're measuring LDL in our typical assay, we are measuring the LDL that's present in LP(a). And so the first important clinical thing to learn about LP(a) is that if your LDL reduction, especially in secondary prevention, we're using high intensity statins, is not reaching 50%, or as you're expected, you might consider that you're measuring LP(a), LDL in the assay. And as you likely know, statins do not reduce LP(a). But, furthermore, we should consider LP(a) as a causative factor in your patient with early cardiovascular disease, but the absence of other traditional risk factors. I have a 33-year-old that has a cabbage, and an elevated LP(a) of 230 is his only risk factor that we can identify. I check LP(a), and I think that this is the practice of the prevention clinic at least once in all the patients that come to me. I think it's important to establish that risk factor, because we know that patients with levels above 125 nanomoles per liter should have more aggressive risk factor reduction. But then you mentioned about potentially being involved in future trials, but and you mentioned this, so I added it about potentially considering that we start a local registry of these patients. So even once treatment is approved, we can be proactive in offering it. And then lastly, this is a larger conversation, but as a system, we need to consider how we're going to deliver these drugs. These are long acting and they're provider dosed. And so, I mentioned inclisiran earlier. We don't yet have a mechanism to prescribe inclisiran, but this is provider dosed, and so those are some sort of clinical implications that we need to think about.
Thanks, Tony. I think that's really helpful. Let's see. Arthur, you want to unmute and give your comments?
Sure. Since we are using multiple therapies to get the LDL cholesterol down to perhaps 60 or less, I think the group that would be interesting to study are those people who have very low LDL on treatment, but have a persistent elevation of LP(a), because this is going to be a high cost treatment. And I think we need to fine tune our therapy to see which patients might benefit the most.
Yes, that's a great point. I think that that population will likely be the group that that we initially treat. Though I'll mention it, though high cost therapy, it seems that the patients who may ultimately derive the largest absolute risk reduction are those who've not yet had an event, right? Primary prevention patients with very elevated LP(a). So I think that it'll be even longer till we have that data, but just to mention that.
Yeah, and just to add that I think, once we start seeing, some of the outcome studies, like with the current phase 3, that will help us to start to understand, kind of the magnitude of added risk from LP(a), because that's going to happen on top of standard therapy. So, I mean, I know it increases risk, but it's been tough to quantify sort of, what is the added increase in risk on top of LDL. So I think starting to look at some of the phase 3 trials when they're going to layer it on top of maximal LDL therapy will help us to sort that out and maybe define better the class of patients who will derive the best benefits from this therapy. And just to sort of, plug or, highlight about inclisiran, and, just to mention that is something that, we've been looking at. Erica Spatz has been working on in terms of being able to get it on formulary for appropriate patients and, to figure out sort of a mechanism for being able to administer it, seeing as it is, as Tony mentioned, a provider dosed or provider delivered therapy. So, maybe more to come on that and how to access it for patients who are appropriate. Other questions?
John Sotaro here. Tony, very nice talk, and it's an important area. As you might remember, in the remote past, it used to be thought that niacin was helpful for patients with LPA elevations. Most of the data hasn't been very good. And at this point, we're pretty much consigned to treating those patients' other risk factors, specifically LDL, as intensely as possible, knowing that the LPA number won't go down at all, which often is frustrating for patients. If people are interested in the Horizons trial, there are a couple sites in New York City that might be useful for patients of ours who are in Fairfield County or Westchester. But otherwise, we're looking forward to the results, and I fully support constructing a registry because there are very many people in this category who might be interested in clinical trials or early clinical use. So I think that's something to think about creating fairly shortly, and it would be pretty easy to do, I think.
Thank you so much, John. I think that's, well appreciated. And again, we'll try to have some of this and share some of this information as well. Jeff, you had a question?
Yeah, Tony, that was terrific. Very, very clear presentation. Exciting stuff. The RNA targeted therapies seem to be very effective in general. It's again, there's a lot of enthusiasm for them. You might have said this and I might have missed it, but what sometimes we're missing is why patients have high Lp(a) levels. I mean, you're describing targeting the RNA so that translation is inhibited and the protein's not made, et cetera, and that's very effective. Are there promoter polymorphisms? Is there something about gene regulation? Because obviously this isn't regulating transcription. This is a post-transcriptional mechanism. So do we know why this tends to be in families and why Lp(a) is elevated and very elevated in some folks?
So at least I don't know. I don't know that's a very good question. And you're exactly right about how we're lowering it. But I don't know that we know why it's particularly elevated in some patients. I will point out something I didn't get into, but let me see if I can go back to that slide. But, importantly, not all Lp(a) is the same. You can see the apolipoprotein A moiety here. The size, the type varies from patient to patient, and there are particular variations of this ApoA that are more atherogenic, more prothrombotic than others. And that's an evolving area of research, but it's likely that those patients are suffering the greatest ASCVD risk and will probably benefit more, but that type of nuanced research I think is to come.
Okay, thank you.
Does anyone else know the answer to this question?
Yeah, let me actually chime in here. So Jeff and others, there are two mechanisms known for elevated Lp(a). One, you mentioned about polymorphism in the promoter region, and there is association. So there are multiple polymorphism, not only that, but also other polymorphisms associated with increase. Second, as Antonio mentioned, The lipoprotein A has the EPO-A has multiple Kringle repeats. Kringle is a pastry, Danish pastry that is rolls around, and that's why it has the shape of what he actually showed to you on this figure. And the number actually matters. So if you have higher number of Kringle repeat, the chance of secreting it from the cell is lower. So patients with lower Kringle repeat are actually at higher risk of having elevated levels in the plasma and also bound to EPO-B. So there are both polymorphisms and the coding region that actually, how many repeats that actually is going to be made in the mRNA that matters.
Great.
Thank you so much, Arya. I think that was a needed input. I certainly didn't know that degree of detail. And just as a comment, Adam mentions that Bridgeport was a site for Horizon, but they've closed enrollment now. So we're just at the top of the hour, and I just want to thank everyone for the participation and very robust discussion in both these presentations today, and to thank again the presenters, Lama and Tony, for doing such an excellent job. And we look forward to seeing you all again next week, same time. Thank you.